Hello scientists, my name is Luis and I'm going to show you a computer program that I developed called LMAPper. This program is very useful to analyze scanning probe microscope images, as you'll see shortly. You can see the program being used. An experimental image can be combined with 3D molecular models. Okay. In this way, analysis and interpretation of the images can be done more easily. But first, I'm going to show you how to get this program on your machine. Open a browser like Firefox, Chrome, or anything you like. Search for LMAPper, okay, and go to the SourceForge link. Okay, this program is being hosted in SourceForge. Now I can have also access to the code if that is something that interests you. Download it. Okay, give a second. We'll save to the desktop. There you go. Okay, here you go. And run it. Okay, some security warning. Just run it anyway. It's quite small, the program itself, but quite useful. Okay, so this is where everything happens. You can import our scanning tunneling microscope or IFM images into this, and can import the 3D molecular images, molecular models onto this together. Okay, I'm just going to show you the same STM image here. Okay, so it supports SXM files from NanoNIS. It accepts STP files from WSXM and some more obscure ones. I'm going to show you this one, which is a format from Nottingham. This one just to show an example. Here you go. Okay, there's some basic functionality here to correct the image. This is the flattening palette and contrast. Type here value. There you go, something like that. Probably okay. Just put on 3. Actually, maybe this one. Okay. Now, as with the molecular models, it accepts PDB files, protein data bank. It accepts also CML, chemical markup language, and also PubChem files, 3D conformers, like this one. Okay, this one, it. In this case, for this image, I know this is a molecular network of PTCDI and melamine. It's something I used to work with. I've got some publications. They self-assemble on a gold surface to form this hexagonal network. The network is supposed to be perfectly hexagonal, okay, where the edges are composed with PTCDIs and the corners with melamines. I can see that I'm moving the molecules around, so I'm basically selecting the molecule with the left click and dragging with the left mouse button. See, so you can pan the molecule around. If you use the right click button, it can rotate on its own axis, quite useful for planar molecules, but you can also do a free rotation using the middle mouse button. Okay, can reset if everything went badly wrong. There you go. Or there is all the functionality, which is the undo. You can use either the menu there, undo or redo. So I'll try now, Control Z. There you go. Okay, and redo. Okay, here you go. So I'm going to put these molecules in the correct places. They should be. Now, there's some functionality here related to the SPM image. Okay. Now I know that my instrument, STM, that I used was not properly calibrated, and this value here should be 1.1. should be about 96 nanometers. That one should be corrected to 1.3. And also that there was some drift. This image was taken at room temperature, so reason why the hexagons don't appear correct. There we go. So this function here, skew, is very useful to correct drifts. If you are familiar with the scanning tunneling microscopy, you will know that there's normally a fast scan direction, in this case X, okay, and a slow scan direction. And that means that normally calibration on X is okay, but any measurements taken on Y will be with an error, okay. And normally what happens that if the tip and the sample drift, you'll end up with an image which is normally skewed, okay. So to correct that skew, there's this function here, okay. So I'm just going to play around a little bit. So let me try to position these molecules. So pan them around. There's a function here to duplicate. Now let's say I want to rotate this molecule by 60 degrees. I cannot type, but I can use the shift button, okay, which will snap to certain angles. In this case is multiples of 15 degrees, okay. Can be useful, okay. Move. I want to duplicate this. Put at the bottom. Oh. Make sure you select when you select the molecule, you actually click on the atoms. Okay. Now, why did I develop this program? I developed this program because in the past, when I want to analyze images like this, I would use programs like Inkscape or CorelDRAW, or nowadays a lot of students actually use PowerPoint. But the problem is that when we import molecular models, we normally import them as a bitmap, and that doesn't allow us to manipulate if we need to. If you want to manipulate the angles in 3D, you'll have to go back to the program where you draw your molecule and take another screenshot, and then you can import. Now this program avoids all that problem, because you can just import directly your molecular models in 3D. Now the other thing is that when you have your molecular model, the information there is also in let's say nanometers, so there's a real space value for the size of the molecules and the position of the atoms. And also when you collect your experimental images, there's also information about the size, and so this program combines all this. It reads that information and puts everything right in scale. Now in the past, sometimes it would be difficult to put everything in scale, and sometimes could lead to errors in measurements. So duplicate this one. Oh, sorry, I can use that one as well. Rotate, and let's do this melamine. Oh, come on. Okay, this probably requires some practice before you can actually use it properly. Okay. Okay, you can see that this molecule forms a triple hydrogen bond. Okay, that's the reason why it forms this zigzag network. This melamine molecule has got here a location where this other molecule the PTCDI can form this triple hydrogen bond, and that's why yeah, it forms this hexagonal arrangement. Okay. Now the one there should be this one. Okay. There you go. Okay. Now, things also useful is that you can save your project. Okay, save. Let's say, okay, I've got some here saved, but let's call this test 1. Okay, so that means that if you exit, you come back, you can load it. My preferred method over loading images or stuff is just to drag and drop, and there you go, everything in the same place. Now there's also another function here called measure, also very useful. Let me just make this bigger. So let's say I want to measure the distance between there and there, those two equivalent atoms. Measure, make sure snap to atoms, so it will go to the center of these atoms. To this nitrogen here. There you go. Distance here and the angle relative to the horizontal. Okay. Also very useful. Okay. Let's remove this. So I've shown you how to import images, how to move them around and manipulate. Kind of a natural way of manipulating things, really. How to save your project and load again. Now, let's say that your image does not work with this program, your STM image or SPM. Now, I can understand that, but yeah, there are thousands of file formats for depending on the instrument you use, and it's pretty much impossible for me to program all plugins for that. Now I programmed some most commonly used ones that we commonly use in our research lab. There's a file SXM from NanoNIS, and also it accepts STP files from WSXM. It's an example here. Okay, an image. Let's say I've got this image from from an SXM file. There you go. It's actually two images. What you can do is to save, manipulate. Okay, so this program here is very good to process the image and manipulate. If you want more functionality, yeah. There's also Gwydion here. You can use that too. Save that as STP. We save to the desktop, and make sure you take a note of the actual size of the image. In this case, 5 by 5. I'll show you why. Actually, for this one you don't need. Okay, so I can import this one here, and there you go. Okay, of course the molecules now make no sense. Okay, can delete them. There you go. Turn on skew. Is actually graphite taken on atmosphere with a. Okay, can also measure without the atoms. Point up to 3.51. Okay, this is calibration problem. Sorry, probably a lot of drift. Okay, in this case. Right. Okay. So what else did I want to show you? Now, very useful as well is the fact that you can import bitmap images. Okay, so JPEG and PNG, or just standard bitmap, I guess. I don't think nobody uses nowadays that. So you can just drag and drop, and yes, there's this window that appears asking for what size is this image. Okay, well, I know this one particularly because I took some notes. Should be 10 by 10 nanometers. Okay, it's important because the JPEG image does not have information about the real size of the image of the microscope that was taken. Okay, so it's 10 nanometers. Now you can import, in this case is an image of a nickel octahedyl porphyrin. Okay, it's got the molecular model here, and there you go. And then you can play around and find out what is probably the best configuration. Okay, this image is not very good. Sorry, you can probably find better ones in the literature. But can provide so this program can provide a good way to see whether your molecule fits onto the image you collect them. Okay, and so on. Okay, I think that's covered pretty much most of this program and how helpful it can be. Oh, there's another feature. Let me not forget that. Let's say you want to export this to PowerPoint. Okay, to present this in your presentation. So you can do that. You just have to do Control-C. Okay. What basically Control-C does is just copies whatever it's on this window to the clipboard. Okay. I'm going to show you PowerPoint. or to Microsoft Word. Okay, it's not just PowerPoint. Well, there you go. Okay, and these areas that are outside the SPM image appear transparent. Oh, I'm just going to show you that. Actually, it's actually quite useful. Oh, sorry, move this. Duplicate. Let's say that one of the molecules spills out of the SPM image. Here you go. And now let's copy. There you go. Delete the previous one and put it also this. And there you go. Oh, sorry, forgot to press Ctrl C. And Ctrl C, Ctrl V. Oh, there you go. See? So you can see that this is actually transparent. If I write some text, and I put this to go back. So sorry, send to front, and there you go. You can see over there. Okay. Perfect. Okay. There's some pixelation. Something cannot avoid because the PowerPoint cannot handle the 3D models themselves. Okay. Maybe in future versions, but for now, use my program. Yes. The other thing that might be useful, but not completely working, is that you can import your images or JPEGs from clipboard. Okay, this is something that I introduced recently. It doesn't work with WSXM. I think if you do Ctrl C, Ctrl V, it appears a dialog asking what the size is, but nothing. Okay, but it does seem to work with WSXM. It's another PTCDI-melamine image. Okay, that's before forming the hexagon network. So Ctrl C and Ctrl V, and yeah, she asks you for the size. Let me go back here. I think it's maybe 20 nanometer. Yeah, 20 by 20. Okay, there's a log function here. That means if you type a value here, same value for the X size, but sometimes it's not the same size. So unlocking. Okay, yeah, there you go. I think you understand that. Okay, and you can play with whatever image you like. Let's put more PTCDI. There you go. There should be one over there, let's say. And oh yes, it's from the same instrument, so I have to rescale this. Slightly annoying. And, depending on the drift, it's normally 5. Sometimes the problem with the pairs is not being perfectly, the electrodes on the pairs not being perfectly aligned 90 degrees. Okay, and it's a combination of 2 here. Oops, maybe like this. Not like this. Yeah, here you go. Okay, so yeah, hope you like this program, and I hope that is useful. And if you do find it useful for your analysis, and if you decide to publish, don't forget to reference it, please. Okay, see ya.